Hi guys, it's Mrs. Shannon, and I'm going to walk you through some of the basics with using the microscopes and procedures for doing the cell survey lab. So we're going to start off really quickly, and we have two types of microscopes. There's the same type of microscope, just two different brands, so you might have the light one or the dark one. They both have the same features, just slightly different. They are light microscopes, meaning that they're going to use the light source from the bottom to be able to see the different parts of the slides that we're looking at. Now, the eyepiece is up here at the top. It's what you look in. And then you have three different magnifications. 1, 2, 3. You can always change it by rotating them around. You always want to start at the lowest magnification, the smallest lens that we have. And it's also a good idea at the end when you're finished with it to put it back in that location just to be on the safe side. Now, the first thing you're going to want to do is get used to using the knobs. Now, on this one, we have a course adjustment here. You can notice, I don't know if you're going to notice it here, that my black tray is going to be going up and down when I move the course adjustment. This is the adjustment that you use for first. It's just to give you a ballpark. When you get it as clear as you can with the course adjustment, then you're going to go down to the fine adjustment and you're going to fine detail it. Once you get it as focused as you can with the coarse and the fine on the lowest magnification, then you go up to the next magnification. You do the same thing with the fine. Only use the coarse adjustment on the lowest magnification. If you need to go, if you feel like it's not focused, it's not working at the next magnification, then go back to the lowest and start again. The reason is this moves the tray up and down. And as the lenses get longer, there's more of a chance to actually smash the lens itself. Now on the middle strength, it's not as bad, but you should never, under any circumstances, on the biggest magnification one, ever use the coarse adjustment. Ever, because it more than likely will smash the slide. Okay? Now here on this one we have 10 times, it's magnified 10, 40, and you'll see one that's labeled, I think it's 5. I think it's 5. Just so you know, a heads up. Now, when you are making your slides, there's going to be a couple different ways that you're going to do it. This is a slide. We also have cover slips that we're going to be using. They are in a white box like this. Please be careful with them. They're very thin. They're easy to stick to one another. And we don't have a whole lot of them, so we need to be really cautious with them. And of course, this one's not open, so we'll open the box up. Okay, some of the items that you will be using will be in liquid form. An example is kombucha. Kombucha is a fermented tea. It's fermented using bacteria, live bacteria cultures, and so it's already in liquid form, and the idea is that we'll see the bacteria, the prokaryotes, this way. Hopefully it doesn't. It is stinky, I will warn you on that. For this one, all you're going to have to do is take one droplet of the kombucha and put it on the slide. And so give me a moment because I haven't opened it. I thought it was going to be a little quicker. It is very stinky. You see it's bubbling up, and that's because of the fermentation process that it underwent. Okay, so all you need to do is take the eyedropper. It should already be in the cup. You probably have the cup by the microscope. And you just need to take one slide and put, whoopsie, one droplet on. If you get 2 droplets, it's not the end of the world. It'll just be a little bit bigger. Now, you need a slide cover on this because what's going to happen is as you're adjusting it, there's a chance of getting the lens wet, and we don't want to get it wet with the stuff because it's going to stay on the lens, and the next time we go to look at a slide, you'll have leftover materials. So I'm going to take one of the cover slips out. There's a little piece of paper, get rid of that. And the way that you put a cover slip on, I'm going to try to get it closer, is you lay it at a 45-degree angle, which is kind of halfway to straight up and down, and then you just let it drop. It helps to get rid of the air bubbles that way. Okay, and then what you're going to do is make sure it is turned on, that the light is showing. You're going to put it on the table, which is the black stand here, take the little brackets and secure it in there. Make sure that the water itself, or the droplet of what you're using, is in the light part, and then you can start doing your adjustments. Once you get it adjusted to what you can see, hopefully you will be able to see something. Always check with Mrs. Shannon to look at what you have to see if what you have is actually something. Then you'll be able to sketch it in your paper. Now the other type of thing that we'll be doing is solids. We'll be doing a lot of vegetables, for instance. And to do vegetables, you need a very small amount. And I don't even know, like I have a piece of carrot, and I'm actually going to use my finger now. I think it gets it better than like a knife, and it's safer. And you need the smallest, can you see that? I don't know, the smallest little sliver that you can get. When you're doing onion, it's super easy because it peels off in nice thin strips. But if you're doing some of the harder vegetables, you'll need to do this. Okay, if it's a solid, you will need to take the distilled water and you will add a droplet of distilled water on top of the solid. And that helps keep it from drying out and the cells from drying out. And then again, you'll put your cover on it just like before, and you'll look at it under the microscope. There will be a couple of items that you will need to stain. What the staining does is it helps you be able to see certain organelles better. I will either have a separate video on the actual staining, or you can talk to me directly and I will show you how to stain in class depending on the amount of time we have. But we have two different stains, iodine and methylene blue, that we will be using, and the instructions in the cell packet will tell you when to use which, I believe. Okay, so I hope this helps get you started. Let me know if you have questions.